So, hi and good evening, everyone, and good morning to those who are joining from US. Uh, we wanted to ask Jeet, will the demo or workshop work fine on phone as well, right? Or they need laptops?
Yes, it should work fine on cell phones as well. You can
Okay.
access GlyGen website and the tools that are there in GlyGen um,
Okay.
on your phone as well.
So we are mentioning that laptops might help for better understanding, but will work on phone also. Yeah.
Yes.
We'll start in another 3 minutes. That is the exact time. So by the time I hope everybody joins. Hello. Good morning, Professor Raza, Professor Michael. Good morning to you and good evening from us here in India. Can we start the session?
Yes, thank you. Good evening to everybody from India and good morning to the ones in US.
Yeah, it's difficult to manage a global session with global audience, but somehow with great professors and enthusiastic audience, we've been able to put this today. So without further ado, let's start. I would now request Govinda, who is the president at RSG India, to please introduce RSG India, and then we move to the session. Over to you, Govinda.
Yeah, thanks, Akriti. And Yeah, without further delay, I will start introducing about the RSG-India, what we do with RSG-India. So RSG-India Regional Student Group, India. So it is your local window to the global opportunities. So in RSG-India, we do the multiple range of activities with the support of ISCB and ISCB Student Council. So let me introduce about the ISCB. So it is International Society for Computational Biology, recognized as one of the global organization for the computational biologists. And it is main aim of this ISCB is to maximizing the global impact of computational biology on science society through the providing scholarships and research training and outreach and inclusive community through the inclusive community. So with the aim of providing this community, it has started the ISCB Student Councils in 2004. So the aim of this ISCB Student Council is to develop the next generation of computational biologists, and it is conceptualized in 2006. And as of now, ISCB Student Council has 26 regional student groups all over the world, and ISCB-RSG India is one of them. So it is subsidiary of the ISCB Student Council, and RSG India aim is to promote the network of computational biologists in India and all over the globe. And as you can see, it is a local window to the global opportunities. And ISCB-RSG India, it is established in 2006 to 2007 in between, and we are here right now. And we are learning from the past experiences, and we are learning from the present experiences as well. We hope we continue for this in the future as well. So, and these are the two recent activities we have done in 2020-24. So one is the Academia to Industry and Computational Biology Career and Advice Tips by the Dr. Tommy, and another creating impact in the science communication opportunity in the computational biology career opportunities. So it is by Priyanka Das Gupta, the communication officer in Geneva. So these are the some wide range of activities we conduct to the RSG India. And in summary, in 2023 and 24, ISCB RSG India has conducted 6 webinars, 5 seminars, and 2 workshops, and 1 biohackathon. So this is our commitment to the RSG India and commitment to share the knowledge through RSG India with your support. And finally, this is our team, myself, Govinda, and Nikita, she is the secretary, and Dr. Manish Kumar, he is the faculty advisor of RSG India, and Akriti, she is the communication advisor, Ashita, she is the nodal head, Sabyasachi, she is the design head present in RSG India. Along with the core committee, we have the several volunteers who work for the RSG India, and you can find us through our socials, and we have our own website and RSG India. You can reach out, you can come join RSG India. And Agriti, stage is yours. Thank you.
Yeah. So today's session is about Meet and Learn with the Bioinformatician. So it is MLTB series. Last year in 23-24, we conducted Meet the Bioinformatician series. It was a series where we requested and reached out to different bioinformaticians across the globe and made them meet with the budding bioinformaticians so that conversations and insightful stories happened. And through these sessions, as Govinda just showed, we had 7 episodes in last year. And this year we have come with a reformed concept that along with the seminar, we also have a workshop or demo so that theory and practical move hand in hand and to multiply learnings and master skills. We are very honored to have the first episode with Professor Raja and Professor Michael Tiemeyer from GlyGen, and we'll know more about GlyGen, Glycans in just a while from now. Next slide, Govinda. So yes, let's have quick glance at a glimpse at the series of 23. We had 7 episodes, about 500 participants, 7 established bioinformaticians, and infinite budding bioinformatician dreams and insightful conversations. Please have a look at last year's events. These were the episodes where we requested and we were honored to have established bioinformaticians across India from various institutes. We also had an episode with Professor Kyoko from Japan, Professor Balram from IIT Delhi, and Professor Alok Bhattacharya from Ashoka University. These were the range of bioinformaticians. And now, after the resounding success of our MTB series in the year 2023, we are humbled to bring back a renewed and evolved version that is Meet and Learn with the Bioinformatician. So let's now move on to knowing the participants of today's event. So today's participants, there were 169 registered participants across the career graph, from undergraduates to postgraduates, PhDs, laboratory technicians, research assistants, and faculty members. We have a wide range of people because learning knows no age. So, and next, these two were the simple questions we tried to ask through registration forms, that were whether you know about glycans, glycoinformatics or glycoscience. And yes, you can see the stats about yes and no. And as you can see, 89% people have voted in this session that they wanted to learn something new. So here we are with our bioinformaticians of today to learn something new. So now I will request Arushi to please introduce them formally. What informally I can say is I know Professor Raja I've met him in person, and all that I can say about him is that he's a great teacher, because he's a teacher who never fails to he's a curious to learn every time, and that's the best quality of a teacher I know. And Professor Michael, I've come to know through the mails of Professor Raja. He agreed to do this session, and we are very happy that he had that enthusiasm. So thank you, both of you. And now I request Arushi to please formally introduce the speakers of today.
It is our distinct honor to introduce our esteemed speakers for today's event. Professor Raza Mazumder is a principal investigator at the George Washington University, US, and the co-PI of GlyGen. Professor Mazumder completed his PhD in microbiology from Virginia Tech and contributed to significant projects at National Center of Biotechnology Information, NCBI, NIH, and PIR Uniprot, a former faculty at Georgetown University. Professor Mazumder specializes in developing innovative bioinformatics methods and leading high-throughput projects such as Biocompute, GlyGen, ARGOS database, and OncoMX, focusing on the cancer biology, glycobiology, microbiology, and machine learning. Professor Michael Tiemeyer. Tiemeyer is a principal investigator at the University of Georgia, Athens, US, state of Georgia, and the director of the Complex Carbohydrate Research Center, CCRC. Professor Tiemeyer completed his PhD in neuroscience in 1989 from Johns Hopkins University. He was a Helen Whitney postdoctoral fellow in developmental neurobiology at the University of California at Berkeley. Prior to joining CCRC faculty, Dr. Tiemeyer was a faculty member in cell biology at Yale University School of Medicine and director of the biochemical and clinical analytics and new methods development at Glycoproteomics. Previously on the faculty at Yale University School of Medicine and director at Biomedical, his research focuses on the glycoconjugates in development and disease, glycomics and glycoproteomics, and protein glycoconjugate interactions. We are incredibly fortunate to have these two eminent scientists with us today. Without further ado, please join me in welcoming Professor Raza Mazumder and Professor Michael Tiemeyer.
So over to you, professors, for the session. Please take forward. And we would also like to introduce that this is a student organization, but we are only being able to conduct all these sessions with the support and guidance of our faculty advisors. Currently, the faculty advisor at RSG-India is Dr. Manish Kumar. And thank you, sir, for joining today. He's there in the call with us. So thank you, sir. And over to you, Professor Mazumder, for joining. Please go ahead.
I think Mike will share his screen first.
Okay, you see my slide okay?
Yes,
Yeah.
Mike, you need to swap your display. We see your not the full screen slideshow mode.
All right, hang on.
So oh, the other one.
How's that?
Okay. Yeah.
Is that
Yeah.
That's
This is
good?
good. Yeah.
Okay, well, thank you for inviting us to participate in this episode. It's a real honor to be able to talk to you all. And Raj and I are going to split up this time in between us, and I'm going to start and try and give you a little bit of background on glycobiology and why you should care about glycans and glycoconjugates. It was really nice to see that many of you are already familiar with glycobiology, glycoscience, and glycoconjugates. So that'll make my job a little bit easier. But hopefully, by the end of my talk, we can all have sort of a common understanding of why glycoscience is so exciting and how informatics is essential, really, to the future of our being able to understand the breadth of the data in this field. So I'm going to start with some sort of basic concepts, and I'll end with just a little bit of a tease into where glycoinformatics is now, and let Raja take off from there. So I'll start with this really essential concept that all living organisms have on their surface a very important and information-rich coating of carbohydrates. If you look here on the left, this is a fungus. Let me get my pointer here. Very simple organism. This is happens to be a yeast cell right here. And this yeast cell is surrounded by this haze, which is all complex carbohydrates. So the capsule that protects this fungus from its environment and controls its interaction with its environment is all complex carbohydrate. So this coating, which is also referred to as glycans or glycoconjugates, are essential for all kinds of biological functions that occur at the surface of cells, especially protection, cell signaling, recognition between cells. There's also glycosylation inside the cell. The so-called nucleocytoplasmic glycosylation is absolutely essential for life and involves the addition of a very simple sugar, N-acetylglucosamine, to transcription factors and elongation factors and all kinds of enzymes that function inside the cell. So this coating on the right here is a human red blood cell, a bit more complex perhaps than a yeast cell. And here you can see the nucleus and the cytoplasm. And then the surface of the cell has been stained so that you can see all the complex carbohydrates. And you can see the surface of the cell in animal cells is also endowed with a very rich coating of carbohydrates. And some people like to refer to this as the glycocalyx, or the coating of carbohydrates on the outside of the cell. So there's a couple of objectives I have in my talk today. I want to sort of share with you my understanding of the scope and the breadth of this field we call glycobiology. I'll talk a little bit about the structural diversity of glycans linked to proteins or lipids and how that diversity arises from how the building blocks are put together. Give you an introduction to the different classes of glycoconjugates that are found in vertebrates. Talk a little bit about their functional impacts and talk also about experimental strategies for understanding how carbohydrates are used in biological systems. And then at the end, I'll just give a brief introduction to glycoinformatics. So about the scope and the breadth of the field. Here's just a listing of some of the major diseases and conditions that are linked to animal glycans. As some of you probably appreciate that our blood groups, ABO, are different from each other because of the carbohydrates that we each individually put on the surface of our cells, especially red blood cells. So A blood type, B blood type, O blood type have different glycans on particular glycoconjugates on the surface of our cells. So glycans and glycan binding proteins are also found on the surface of inflammatory cells, such as neutrophils, eosinophils, and macrophages. And there they are very important for regulating inflammatory responses, how severe an inflammatory response is, for instance, and also controlling autoimmune responses that are very important in asthma, irritable bowel disease, emphysema, rheumatoid arthritis. So glycans and their recognition by glycan binding proteins regulate the immune response and inflammatory responses. Cancer cells have their own frequently unique sets of glycans on their surface. And as cancer cells become metastatic, that is, as they begin to leave a tumor and go to other parts of the body, they frequently change the carbohydrates that are on their surface. And this allows them to actually escape one place and move to another. So thinking about the whole field of glycobiology and glycomedicine, if you will, we know that altered glycan expression has impacts on a lot of different diseases. And actually, glycans are therapeutic targets for some diseases. And these diseases include diabetes, muscular dystrophies, storage disorders such as mucopolysaccharidoses, neurodegenerative diseases like Alzheimer's, X-linked intellectual disabilities, sickle cell disease, blood clotting difficulties, stroke, host-pathogen interactions, whether that's viral or bacterial. All of these involve glycans in one way or another. So there's also, though, a role for glycans just in normal tissue architecture and normal structure and function of tissues. And so again, here we come to the glycocalyx to think about and sort of illuminate what these roles might be in normal tissue function, right? So you start by understanding that the glycocalyx that I mentioned to you, the composition of that, the carbohydrates that are in that change. It's a dynamic structure, and the actual composition is regulated and can be very exquisitely varied from one cell to another. And here's one of my favorite examples of this. This is a rat brain that's been, a section of a rat brain that's been stained with 3 different antibodies. And each of those antibodies bind to the same protein. So we're looking at the distribution of this particular protein across the brain, but the different antibodies stain different glycoforms of that protein. That is different, the same protein with a different carbohydrate on it. So we're seeing how one particular protein is glycosylated differently in different regions of the brain. So you can see in the brainstem, there's a lot of this blue form of this protein. In the pons, there's a lot of the red form. And if you look up in the cortex here, and higher magnification on the right, you can see that the same protein expressed in 2 neighboring cells can be glycosylated in different ways. So this really sort of poses some important questions for us to sort of think about. What are the functions that are subserved by this very, very tightly regulated cell-specific glycan presentation? Not just at the level of the tissue, but at the level of cells that are next to each other. What are the functional consequences of that? Also, how does this finely regulated glycan expression kind of fit with what we think of in terms of the, if you will, the central dogma of biology, which if you distill it down, it really says that regulated gene expression controls biology. Here we're looking at not at regulated gene expression, but regulated carbohydrate expression. How does that fit into what we like to think of as the central dogma? So just to remind you, the central dogma that grew out of our understanding of the structure of DNA, really, and how DNA is replicated. So the central dogma states that DNA encodes RNA directly, and RNA in turn can be translated into proteins. And then if you sort of extend that, we think, well, there are proteins that can feed back and control the transcription of RNA from DNA. So proteins control the production of themselves. Proteins work together to form cells, and which are larger organizations of proteins and molecules, and cells interact with each other to form organisms. So that's sort of the central dogma, right? But that really, really ignores a role for these other very important sorts of molecules like lipids and carbohydrates that are found at the cell surface. And so maybe a different way, I don't know if those, am I adding those lines or not? But another way to think about this central dogma is, thanks, that DNA encodes RNA, encodes protein, but many of these proteins are enzymes. And these enzymes can build things like glycans and lipids, making glycoconjugates. The glycoconjugates that are found in high abundance at the cell surface of cells inform the interactions of cells with each other to become organisms. So you see now that carbohydrates are spread across the whole central dogma. In fact, if you want to get really picky about it, DNA and RNA are actually just sort of complex carbohydrates themselves that have ribose as their backbone. But the point is that really carbohydrates feed into the central dogma and modulate how organisms function. Carbohydrates are also very essential now as therapeutics. And this is a bit of an old slide, but it makes my point, I think, very well. That is that many of the newest therapies that are being developed for all kinds of diseases have arisen out of our ability to make proteins in host cell manufacturing systems like CHO cells and other cell types. Almost all of these therapeutics are broadly called biologics, are glycoproteins. You think of all the antibodies, the monoclonal antibody therapeutics, those are glycoproteins. Vaccines are many times are complex carbohydrates of bacterial cell surfaces. So this really amazing growth now, well beyond, this is only up to 2017 in this market for biologics, requires understanding something about how proteins are glycosylated and what those glycans do to the function of the protein. And this is really the first example of a good understanding of why this is important. So this drug, erythropoietin, was one of the first biologics made by a pharmaceutical company. And erythropoietin, you may know, is a hormone that's responsible for stimulating the maturation of red blood cells. So in applications like chemotherapy for cancer, where red blood cell production is reduced, erythropoietin can help reconstitute the important level of red blood cells in a person. But erythropoietin is a relatively small protein. You see it here in blue, it's 18 kilodaltons. Then all of this red stuff attached to erythropoietin are glycans, the so-called N-link glycans. And erythropoietin, as you get it out of a bloodstream, has 3 N-link glycans and usually 1 O-link glycan, which really almost doubles the molecular weight of this protein. If you express this protein in CHO cells, like Amgen did to make their drug Epogen, it comes out of those CHO cells with 3 N-link glycans and 1 O-link glycan. And so here's sort of the natural history of that drug. Erythropoietin was introduced, and this is the sales of the drug over the years. Became a very popular drug in the early part of this century, really good sales. And then Amgen started to mess around with erythropoietin a little bit to see if they could make it an even better drug. And what they did was add additional N-glycosylation sites onto the backbone. So here's the native Epogen, erythropoietin with 3 N-link sites, and we're looking at the dose of the drug on the x-axis that's needed to produce red blood cells as measured by iron incorporation. So it's a pretty good drug. But as they added N-link glycan sites, here are now 4 sites, 5 sites, and 6 sites, the potency of the drug dropped by orders of magnitude, right? So from a business standpoint, they can make with less materials, they can make more money, they save on cost of materials, and they have a lower dose to give a patient, which is usually very efficacious. So that when that drug was introduced, it became a real blockbuster, and sales absolutely went through the roof. And for our purposes, really pointed out that understanding how your protein is glycosylated has a big impact on understanding its function. So I'm going to talk a little bit now about some basic terms in glycobiology and how glycans are put together. And so some simple terms, basic terms, monosaccharide is simply a carbohydrate that cannot be broken down into anything smaller by treatment with acids. So this is a simple sugar, a single building block. Oligosaccharides are usually 4 to 12 of these monosaccharide units. Polysaccharides are usually anything greater than 12 monosaccharides. And polysaccharides usually imply that there's a presence of a repeating motif, maybe disaccharides that repeat and repeat and repeat to form a long chain. Okay, different types of glycoconjugates. Glycoproteins obviously are protein plus carbohydrate, and I've already mentioned that most of the proteins of biological and therapeutic significance are glycoproteins. Glycolipids are lipids plus carbohydrate. These are very important markers for different types of tissues. And also many glycolipids serve as receptors for pathogens. Most of the blood group antigens are found on glycolipids in highest abundance. And they've also been identified as tumor-associated antigens as diagnostics. Proteoglycans are kind of like glycoproteins. They are glycans that are attached to a protein, but in this case, the glycan itself is a large complex chain known as a glycosaminoglycan. These have very immense structural diversity and have a lot of different functions, various developmental pathways and blood clotting pathways. So glycoproteins, glycolipids, and proteoglycans are all built really from the same building blocks. There's a relatively small number of monosaccharides that are important for building vertebrate glycans, Glucose, I'm sure you're familiar with. Glucose, galactose. Note that the difference between glucose and galactose is simply the position of this hydroxyl. In glucose, in this formation, it points down. In this formulation, it points up. They're the same chemical makeup, same number of carbons, same number of hydrogens, same number of oxygens. The only thing that distinguishes one from another is the stereochemistry. That stereochemistry is utilized to build a lot of different structures. So you can link a galactose to a glucose, for instance, from this position, the one position of the galactose, to this position of the glucose, this position of the glucose, this position of the glucose, or this position of the glucose. And you end up with a disaccharide. If you link that galactose to the 4 position of the glucose, you end up with a structure that's known as a milk sugar or lactose. But you can also link the galactose to any of those other hydroxyls to build different sorts of molecules. And in fact, if you think about linking building blocks together, for proteins and nucleic acids, if you take 3 amino acids or 3 nucleotides, there's only 6 ways you can link them together because they are a linear polymer. So there's ABC, there's only so many ways you can link that together linearly. But because carbohydrates can be linked in different positions, you can take 3 different monosaccharides and link them together between 1,000 and 27,000 different ways, depending on whether you restrict yourself to what biology actually can do because it has the enzymes to do it. But chemically, 3 monosaccharides can be linked together in 27,000 different ways. So there's a vast potential for encoding bits of information in the structure of a carbohydrate. And on top of this ability to change how they're put together, frequently after they're put together, they get modified again with other substituents like sulfate and phosphate, which increases the structural diversity even further. So something about carbohydrate synthesis, because carbohydrates are not built, they're not encoded by a linear program, a linear template like DNA or RNA, they're considered secondary gene products. They're built by enzymes. They're not encoded in the DNA. They require, for the correct synthesis, enzymes to put them together, glycosyltransferases, enzymes to take them apart, glycosidases, and the availability of all the substrates that need to be there together in one place to build the structure. So that means the distribution of organelles inside of a cell regulates how glycans can be assembled because all of these pieces are found in organelles within the cell. So a consequence of this is that glycosylation can be very sensitive to changes in the environment that influence substrate availability, that influence energy levels in the cell. So glycosylation frequently reflects something about the environment of that cell. Some other important basic terms that is good to have in your toolbox as you think about informatics of glycans. The term microheterogeneity refers to the fact that on a single glycoprotein, there can be different glycans found at each of the glycosylation sites. So one particular glycosylation site may be unused or used, may have a glycan or not, and that glycan can come in different flavors. So a single glycoprotein can be found in many different glycoforms as a result of this microheterogeneity. And I introduced to you that concept of glycoforms when I showed you that rat brain. Another term you'll hear sometimes is glycone versus aglycone. So the carbohydrate portion of a glycoconjugate is called the glycone. The protein portion is considered the aglycone. That's really a chemical term, but you see that in the literature every now and then. So I'm going to introduce you to some of the classes of glycoconjugates that are found in vertebrates. I told you about glycoproteins, glycolipids, but I'm going to zoom in a little bit, talk about the specific diversity of some of those structures. So this is sort of a bird's eye view, if you will, of a cell. Here's the plasma membrane. This will be inside the cell, outside the cell. And outside the cell in that glycocalyx, you see different types of glycoconjugates. So one that you would find really close to the membrane that may actually serve as an anchor for a protein here, shown in black, is a glycophosphatidylainositol or GPI anchor. So this is a series of glycans linked to a lipid that forms the bridge to a protein and keeps that protein attached to the cell surface. So the GPI anchors. On glycoproteins, you find N-linked glycans. We'll look at these a little more closely in a second. They come in different flavors, complex, hybrid, and high mannose. There are also O-linked glycans that are found attached to the protein through oxygen atoms. N-linked glycans are attached through nitrogen atoms on asparagine residues. O-linked glycans are attached through serines and threonine hydroxyls. And if they begin with this yellow square here, they're called mucin type, and they come in different shapes and different flavors. There are also other types of O-link glycans that are found on specific structural folds on proteins and help those folds form. So there are domain-specific O-glycans and mucin-type O-glycans. There are also the glycosaminoglycans I mentioned, which are these long polymers that can be sulfated, are very structurally diverse, as well as one lone example of a free soluble glycan called hyaluronic acid, which looks very much like a glycosaminoglycan, but it's not attached to a protein. I also mentioned that there's glycosylation inside the cell, in the nucleus and the cytoplasm, and this is O-linked N-acetylglucosamine or O-linked GLCNAC, and it's found on many different types of proteins in the cell. And of course, you're familiar probably with glycogen, which is found on a protein called glycogenin as a storage form of glucose. Collagen also has its own unique set of glycans, very simple glycans, but they're essential for collagen's ability to fold into the helix it needs to fold into to be a good structural protein. And then finally, there are the glycolipids, and here's a few representatives where glycans are attached to a lipid, and in this case, a ceramide, not a phospholipid, and are found also at the cell surface. So I want to point out here that these shapes that I'm showing you have meaning, in both informatically, but also to glycobiologists. There is a code here, and this has been harmonized by the as a symbol nomenclature for glycomics. And these shapes and these oligosaccharides can be encoded in machine-readable formats using specific descriptors. And each of these glycan structures also has its own unique identifier, a glytucan ID, which allows it to be referred to in a machine-readable way. So a little bit about the different classes of glycoconjugates. We're going to start with N-link glycans. And this is a really messy figure, but I want you to sort of focus right here on this bit of the central dogma where a messenger RNA is being translated into a protein. That protein is being inserted into the endoplasmic reticulum. And if that protein has a consensus of an asparagine, any amino acid except proline, and then a serine, threonine, or cysteine, that asparagine can be modified with this big glycan that is sitting in the endoplasmic reticulum on a lipid called a dolichol. So the nascent protein, as it's being translated, can be glycosylated if it has this consensus. And this is a very studied consensus and a very strict consensus for this type of glycosylation. I want to point out in this whole other bit of mess of this figure, simply that a lot of things impinge on the ability to do this glycosylation reaction. Glucose, so energy metabolism, lipid metabolism to make this dolichol, all of these enzymes, all kinds of components of normal cellular function have to come together in order to make this glycosylation happen. These are the major classes of N-glycans, and oligomannose, which has a lot of these green circles, so there's a lot of mannose. Complex glycan, which has removed many of those mannoses and replaced them with what we call complex arms that show that this glycan has been processed. And then hybrid glycans that retain a bit of that high mannose character, but also have a complex arm as well. So all this great diversity, does it have any meaning? Well, I'll show you probably one very simple example. If you make a mouse that can't add this blue square to this arm, so it can't make the complex structures, it can't make hybrid structures, it only makes oligomannose structures, that mouse is dead. And that's shown here. This is a normal mouse embryo at day 13 of gestation. This is a mouse that lacks that enzyme, and it doesn't survive past mid-gestation. So yes, N-glycans are essential for life. O-glycans, another class of glycans linked to serines and threonines. I'm showing here the mucin type. They all start with this yellow square, which is an N-acetylgalactosamine. Are frequently found on mucin type proteins in high concentration on concentrated into stretches of the protein. And they come in different shapes and different families that we can identify based on how that core disaccharide is modified. There's no real strong consensus for whether or not a serine or threonine is going to get modified by glycosylation. Frequently, they're found in patches of a lot of serines and threonines with prolines interspersed, but it's not like N-glycosylation where there's a very clear consensus. As I mentioned before, there's also some other types of O-glycosylation, the addition of mannose, the addition of fucose, the addition of glucose. Are O-glycans important? Well, they're important enough that some companies have been able to raise substantial amounts of funding to develop drugs that target the O-glycan biosynthetic pathway, or O-linked glycans, especially related to cancer, where O-linked glycosylation has been shown to change in very interesting ways, providing targets for new therapeutics to kill cancer cells. Just briefly, glycolipids. I'm showing you here the full class of the gangliose series of glycolipids, which starts with a ceramide here, and that ceramide gets a glucose added to it, and that glucose can be elongated in many different ways to form this whole family of glycans, all linked to a lipid called ceramide. Are these important? Well, if you knock this enzyme out in a mouse, it's an enzyme that adds this purple triangle to this yellow circle. It's a mouse knockout of this ST3-gal5 enzyme, it's called. This mouse is viable. It's resistant to type 2 diabetes. It's enhanced insulin receptor signaling, but it does have some loss of hearing and some fertility issues, but otherwise it's perfectly viable. So that's kind of strange, because everybody thought these lipids were going to be really important. Turns out mice have a workaround, and if you're a human and you don't have that enzyme, you're in bad shape. There's been 2 cohorts in the United States now identified of patients that have reduced activity of that enzyme. They have profound intellectual disability, failure to thrive, seizure disorder, and some structural problems as well. One cohort in African-American community in South Carolina, and another in the Amish community across the United States. So I'm just going to spend a minute or two and talk about strategies for understanding how glycans might function. And this refers to the fact that we have glycans on our cell surfaces, but we also have proteins that recognize those surfaces, and, if you will, sort of translate the glycome. So one cell has a glycan on its surface that might be recognized by a glycan binding protein on another cell, and that recognition may allow one cell to see the other cell as being self, as being part of the organism, an appropriate partner. Well, it turns out that microbes have recognized that we do this, and microbes also put glycans on their surfaces to try and look like self as well. Microbes also have their own glycan binding proteins that they use to interact with us in host pathogen interactions. So these glycan binding proteins allow us an inroads into understanding something about the function of glycans. In fact, the binding specificity of these glycan binding proteins allows us to sort of test hypotheses regarding glycan function. So the more we understand about the specificity of these glycan binding proteins, the more we can understand about the function of those glycans. And so, so here's an example, for instance, of vascular endothelial cells that may have glycoproteins on their surface that are recognized by glycan binding proteins on circulating leukocytes, and then that can result in changes in cell signaling. And we know that these interactions can mediate cell adhesion. The signaling can mediate cell death or cell division and regulate cell migration. This has been very well studied in immunity and inflammation. But over the last couple decades, a lot of data has been generated in the field by using what are called glycan arrays. And so these are simply spotted arrays of glycan structures on a glass slide that then can be probed with glycan binding proteins that have a fluorescent tag, right? So you generate data about what glycan binding proteins recognize what glycans. And of course, this binding protein has an ID. So you got a uniprot ID, and these glycans have IDs. They have glytucan IDs. So you can begin to see how we can build up data sets with good identifiers that can allow mining and building hypotheses about binding specificities and binding interactions. And we know now that it's frequently the external bits of these glycans, so-called structural motifs out at the end, that are frequently what's recognized by these binding proteins. So I'll just finish up here with 2 slides, just a little bit of context for glycoinformatics. And I told you that glycoconjugates are found in almost every compartment of the cell, especially at the cell surface. And whether they're secreted or found at the cell surface, they can regulate disease progress and important for health and tissue function. Almost all of the currently marketed biologic pharmaceuticals are glycoproteins, and the glycan structural features can frequently influence the function of the molecule to which they're attached. But where's all this data captured, and how can we harvest it in a way that's informative and useful to biomedical researchers? I've also told you that there are some harmonized representatives for glycans and for proteins, of course, and they're machine readable ways to describe these. And we know, of course, that there are great resources for some of the major building blocks of life. For nucleic acids, we have good repositories for proteins, for lipids. But for glycans, that's been missing. Where's all the data from glycans that define what our glycomes are? Where are these databases? Well, we're beginning to see them develop over the last 6-7 years. And Raja will tell you a bit about GlyGen, but there are also other efforts around the world, like Cosmos in Japan, GlyConnect in Switzerland, and together we work with these groups to form an alliance called the GlySpace Alliance to make sure that we're all using the same ontologies and we're all moving forward together so we can intercommunicate with each other. And I'll just finish with this slide, just reflecting on some opportunities for understanding glycan functions by being able to present our data in a way that other people can use our data and we can use other people's data. And things like thinking about gene expression and biosynthesis and how that interrelates with glycan biosynthetic activity. So can we compare gene, protein, and glycan expression changes across normal and diseased states and begin to understand co-regulation of genes and glycans? Through understanding glycan binding protein specificities, which I mentioned, which may allow us to understand how outcomes, good or bad outcomes in specific biological contexts, especially related to inflammation and other disorders regarding autoimmunity, especially in neurodegeneration. Connecting with metabolomic data to understand how metabolomic state, how energy state of a cell, how nutritional state of a cell might regulate glycosylation in good ways or bad ways. So there's a lot of types of data out there that we can connect with if we can serve our data up in a way that's usable across different domains of knowledge. And I will stop there and turn over to Raja.
All right. Thank you, Mike. I'll just go right into my talk. I will go really quickly over a lot of stuff, but all of the data that Mike showed, that data needs to be captured, right? I mean, majority of you are in informatics and bioinformatics, but without understanding the bio part, there is no informatics. So what Mike was showing is really important. But before we start, in the chat message box, you have this link. I would urge you to bookmark this link because that's a wiki page link. And from there are other links that you may use right now during my talk or later on. So let me share my screen correctly. Give me 1 second. All right. So this is where I am. I'm at George Washington University, and this is the metro station right outside Foggy Bottom. And this is the entrance to the building my office is in, very close to Kennedy Center. If you ever been to DC, or if you ever come to DC, you know, this is really nice, just right by the river, Potomac River, and there's a really nice path and walk near Kennedy Center. So lots of people involved in the GlyGen project. I'm not going to go over everybody's name, but I want to highlight Jeet. You know, Jeet and Renee, they are on the call today. And there are others who have been working with us. And we also have our advisors. And I also want to acknowledge ISCB, RSG-India for organizing this talk. And also some of the slides are from Dr. Darren Natale and the annotation slides. If you are all on the link, you will find that there is a feedback form. You don't need to do right now, but in that link, I would really appreciate if you can send us some feedback. And also we have a use case collection form. So the biggest challenge is that when you build an informatics resource, you know, we are engineers, we are bioinformaticians, we build it, but we also want to know what users want and need. And depending on what the users need, you know, we will always try to improve. So there are a couple of proteins I will talk about today. One is the interferon gamma receptor, and the other one is EGFR. And these are the links to those proteins. Before I get into all of that, you know, we were talking about function, right? So Mike talked about a lot of, or gave a lot of evidence where glycans are important for function. You know, the organism may not survive, or if they survive, there are issues with the organism. So it's really important. So just looking at DNA and protein is not the end all of the story. But to look at this, we also know, I mean, I will be talking about glycoconjugates of protein, the glycoproteins, but looking at it from the protein angle and the glycan angle together can help us understand. So if you look at the sequence, proteins and the DNA sequences, you know, all organisms can be divided into bacteria, archaea, or eukarya. Of course, you know, they're the viruses, but in cellular organisms, these are the three major groups. And Carl Woese in 1990, he identified these major groups, and it stands, and this is how we are looking at a lot of biomolecule data, you know, which organisms they are present in and how they work. And I always have Dr. Margaret Dayhoff in my talks like this, because she's a pioneer. She was a pioneer, and she's the one. A lot of things that you are learning in class or you have learned in class started with Margaret Dayhoff and her colleagues. So this was the first database. Well, it was a publication, Atlas of Protein Sequence and Structures in 1965. And she was a physical chemist and a bioinformatics pioneer. And she's the one who developed the PAM matrix and other matrices that you use when you are trying to do sequence-based alignments. So this is how a matrix looks like. It's in all bioinformatics books, if you have you know, seen them in lectures or classes. So when you align sequences, you can ask questions, is that what are the sequences that align and what are the sequences that seems to differ between different organisms? And then Janet Thornton, you know, 1994, and many, many others has, you know, improved upon our understanding on how sequences evolve, protein sequences evolve. This is a BLAST is a tool. Again, you can't talk about bioinformatics with some of the major tools. It's, I think, still one of the largest cited papers ever, right? In the top 100 cited papers. So BLAST allows you to look for homologous sequences in databases. And this is an example of a BLAST results where you take a sequence, you know, I took a UniProt accession sequence here, and then I blasted it. And you can see that in different organisms, there are at least two domains in the protein. In some organisms, there's only one domain present. And by looking at BLAST results, just by looking at the BLAST results, you can tell. So if you have a hit in your BLAST results from animals and plants and yeast, that protein has been around 1.7 billion years. So I can look at a BLAST result and I can tell you, or you can tell, if that protein is essential or not. If you knock out that protein, then the organism most likely won't survive because that protein has been around for that long. So looking at some of the glycosyl transferases, for example, one can ask the question, how many of them have been around for how many billions of years? And try to understand the role. So trying to come at function from a different angle by looking at the glycosyl transferases and glycohydrolases to see how important they are. So when you look at these things, then you start putting these together from a database perspective. That's what we are interested in GlyGen, is that how do you capture this information in a structured way in form of a comment or explanation? So the dictionary, Merriam-Webster dictionary, says is a note, is a comment. Annotation is a comment or explanation. So for genomic data, it can be position of genes. For genes, it can be position of introns and etc. For glycans, we can have name, function, role, classification, interaction, and so on. So the concept of function, there's no one simple answer. What is the function of a biomolecule? It's a very complex answer. So trying to understand all these things for a protein, it's easy to do at some level. Something which is known is experimentally characterized. For glycans, it'll be the same thing. If something is known and experimentally characterized, then you know it's known. What is obvious? Close similarity to other characterized proteins. Non-obvious is when you do BLAST or other sequence analysis tools, it's the alignments are really not very good. Only, let's say, 10% of the amino acids are identical, or less than 10, 5%. What do you do with those things? So those require bioinformatic analysis. And there are several publications which talks about it, including from our group, how to deal with these kind of things. And then the unknown. There are protein sequences which are conserved, seems to be there in all organisms, but nobody knows the function. So if you're starting a PhD or a postdoc, maybe that's something you would want to look at. What are the conserved protein sequences? We really do not know what the function is. So I'm going to jump to a paper in Nature Genetics in 2005 that, stimulated me. Just like you, I was reading papers, going to conferences and so on. And this paper showed that there were 3 children which had the same missense mutation, which resulted in a gain of glycosylation. And this paper also showed the bioinformatics angle of it. So the authors in this paper went and used HGMD, which is a proprietary database, but a really good database, where they looked at the mutations and say, okay, how many of them are resulting in gain of glycosylation? Remember, Mike showed N, X, S, or T. So if there is an asparagine that is gained, maybe that sequon will result in a gain of glycosylation. So there are lots of these kind of mutations, polymorphisms, which results in gain or loss of glycosylation. And then fast forward, when I was preparing for a talk, actually for the talk that I did in Delhi, India, sometime back, and there was a paper in 2019, which showed the structural basis of that gain of glycosylation, why that particular gain of glycosylation results in change of function. So when you do bioinformatic analysis, maybe decades will go by before it is proven or shown, what exactly is happening. But that's the fun part. Papers that you write now, maybe 5-10 years later, somebody will show experimentally why that is important. So looking at from a database angle, what we have here is a protein sequence in GlyGen. And in GlyGen, in the protein sequence, there is a Glycan attached to it. So we can see that there's a Glycan attached to it. The question mark means for the experimental knowledge, it's not known what kind of linkages there are, but majority of the linkages might be known. And on the left side, you can choose to see what are hexoses or mannoses and so on. So this link will take you to that page. And when you are thinking about understanding function, just looking at the Glycan or a single Glycan or a protein will not tell you much. You really have to have data integrated. You have the data harmonized and integrated. So you have to have a data model. So our high-level data model is we have a protein, which can have a glycan attached to it. And then on the glycan can be attached in a particular site in the protein. So the main important things is that we need to have a protein accession, a glycan accession, and a position, a concept of a position in the protein. Once you have these three things, then you can attach a ton of other things. You can have gene expression data on it, function of the protein, glycan, you can say what subtype it is, what if there is phosphorylation, if there is variation, is that a biomarker? All kinds of things can be attached. And once you have these attached things, you can do Boolean searches, knowledge graph-based searches, and other types of things. So the other thing is that when you go to a database, the first thing you need to ask yourself, what does a database have? So many databases may not have everything in the world, right? As generating data becomes easier and easier, what all resources are trying to do is that figuring out how best to show the data so that it can allow people to use their data to map to what is already shown. So in GlyGen, we are adding organisms one by one. We have the fruit fly yeast, Dictyostelium, pig, chicken, Arabidopsis, human, mouse, rat, and others. And we are slowly adding more organisms to GlyGen because when we add something, it's something we want to make sure we are comprehensive enough. So how do you enter GlyGen? You can type in on Google GlyGen space EGFR. I sometimes do that. But Google has its own, your particular Google search might result in a slightly different result. Or you can go to the GlyGen webpage and type in EGFR in the search box, like I show on the right-hand side. And once you go, and by the way, these screenshots are mimics a cell phone. And the reason I do this is that many times I believe resources need to be aware that we are in a train or a bus, or we are walking somewhere to a meeting or sitting in a meeting. We don't have our laptops in front of us. So we take extra attention to make sure that our web pages are mobile friendly. So in this example, EGFR search, there are 682 results. You can filter those results by binding, disease, pathway, and so on. And then from there, you can click on a particular entry, P00533-1, for example, and you can go to the glycosylation section of that entry. And when you click on it, the glycosylation, it tells you how many sites are glycosylated. It tells you where the data comes from, because we really want to provide the evidence for the data. You can download the data as a table, and you can parse it, and you can, after you have it on Excel or something similar, or Google Sheets, you can also do your own other searches, or you can write macros on it, or even, TSV, CSV files. You can do a bunch of things with it. And we are not isolated in GlyGen from the rest of the worldwide resources, right? So that is the key. If you ever build a database or a resource, do try to make sure that they are well integrated. Here we are showing here, I'm showing here how we are connecting to UniProt. and then on the right-hand side of this page, you see a UniProt entry. And from the UniProt entry, you can get more information. So one resource may not be able to answer all your biological questions, but the goal of the resource is to make sure that you are able to pull out the kind of information that you need. Another way to enter GlyGen other than a search by a protein name is GlyGen BLAST. So you can put in the accession number. This is the EGFR accession number. If I put it in and then retrieve the sequence and I click on BLAST, it will pull out the sequence. And if I click on the sequence, I can look at the alignment, not the sequence, actually these two small dots in blue. It will show you the alignment. And on the right-hand side, this is pretty unique for GlyGen. Sorry. Okay, I'm almost done. So the sites and the other important thing to look at function is sites with glycosylation sites plus mutagenesis data. So you can go to a site search and I put here, I want all proteins which has asparagine, and that asparagine has site annotation for a glycosylation and mutagenesis data. This is really important. So there is data to show that if you mutate that particular amino acid, let's say that asparagine, then they have some functional information. So this is one way to show, and I pulled out, there's clustering, and then I look at one protein, and in this protein, we have papers associated with this protein in GlyGen where you can see they actually show that the binding increases by threefold in the paper when the glycan is not present. So this is another way to look at function of glycans if it is present on a protein, which is the glycosylated version of the protein versus the one that is not glycosylated. This is a site view, and this is a very high level view of a super search. It's kind of a knowledge graph. It shows the number of glycans, how many organisms we have, 11 organisms, the number of genes, the diseases that are connected, which are related to the sites, the proteins which have glycans on them, the enzymes which attach those glycans. So this kind of knowledge graphs allows you to visualize what is in the database. And you can do searches by clicking on each of them and filtering the results, just like you do in Expedia or Make My Trip or whatever website you use. So with that, you can get in touch with us if you have a question. You can be under help. There's a contact page, and you can ask us questions. With that, there are few questions that was already sent to us. We have only 3 minutes left, but we can try to answer as much as possible of those questions. So the first question that we have, Mike, you want to take the first question? It is, what drives the formation of glycans in virus coat proteins?
Yeah, so for any virus, the glycosylation that virus shows on its surface, it will reflect the glycosylation machinery of the host cell. So whatever that host cell has as its machinery is what the virus is going to use, but viruses do seem to be able to change the trafficking through the secretory pathway in some ways so that glycosylation can be skewed. And it's usually a combination of altering the secretory pathway, or the viral protein itself has some structural features that only allow certain types of glycans to be presented on its surface. But again, it's still using the host cell machinery.
All right. And I will answer one question. There are several questions, but we can send responses later on, Akriti, to the questions that we cannot answer today, or we can stick around. So one question is about a career path in glycoinformatics. So the way I see it is that, yes, for many students that I also teach, what I find is that knowledge of the biomolecules, the DNA, the protein, the glycans, the lipids is really, really, really important. Can't emphasize. Yes, I know Python and C++ and all these things are all great. But end of the day, if you do not know if you cannot ask questions, if you already always have to be told what questions that you should ask, that's not going to work out in the long run. You'll get to the first job. So I always recommend to my students, try to understand why are these biomolecules important? And then when you are familiar with it, of course, pick up other languages and programming languages. But the type of jobs is big data. So the amount of data is large. Understanding how to organize, handle, write programs which can parse terabytes of data is going to be more and more important. Other part is interface development, understanding what the users want. So those are some of the things I can see. PhD also, there's a big need of people who do PhDs in these fields, informatics fields. So those are some of the things I can say. Mike, you want to add anything to that?
No, I think you covered that well. Is there another question we should address here? Let's see.
Some of the questions we already answered, that we do work with NCBI GenBank.
And there is a place in the central dogma, I hope, for Glycan.
Akriti or Govinda, any particular question that we should close the session with?
So if you have 10 more minutes, could we like cover the questions that we have in the chat box?
Oh, yes, sure. I have. Mike, you good?
Okay.
Yep, I'm good.
Thank you. Nikita, are you there? I request you to please take the questions. As yeah, by the time Nikita. Okay, she's unable to unmute herself. So in the interest of time, if Jeet is available, I request you to please unmute her for the next part of the session.
Yes.
So by the time I'll ask the question. So Ashwin is asking, how can I interlink the network pharmacology with those phytochemical compounds which comes under gene which has more than one target? I think it is a more drug-related question, and maybe if you would like to take up, Professor Mazumder or Professor Tiemeyer. How can I interlink the network pharmacology with phytochemical compounds, which comes under the gene which has more than one target? So probably we would appreciate more glycans related questions at the moment.
Well, I mean, there is one possible answer to that question is, of course, we know what the genes are that control glycan production, the enzymes, and if you have any changes in the expression of those genes as a result of a drug, that's one way to link towards glycan structures through the genes that encode the enzymes.
Okay, thank you. I hope, Ashwin, you've got your answer. Aditya is asking, what could be the role of epigenetics in glycan binding and tumor-associated antigens responsible for the proliferation of tumor cells? Have you gone through any such studies or?
So I can point to one maybe fertile place to look. So I mentioned this phenomenon of nucleocytoplasmic glycosylation, which is the addition of O-GlcNAc onto these proteins, especially transcription factors in the nucleus. And a lot of these, not just transcription factors, but also DNA methylation, all the sorts of pieces of epigenetic, biochemical epigenetic changes are tightly regulated by whether or not those proteins have phosphate on them or have O-GlcNAc on them. So the epigenetic components that contribute to changes in cancer cells are themselves frequently regulated by these glycosylation reactions that happen on nuclear and cytoplasmic proteins.
Okay.
If that helps.
Yeah, thank you so much. We have just 2 more questions in the chat box, and I request Nikita to please take over from here.
Mike, can you see the
hello,
question?
sir.
Okay.
Hello, sir. So this is one of my questions. I think this is not related to glycans basically, but since you have shown that database also, like there should be some parameters that we showcase in a database. So there are so many parameters related to particular thing, like you have shown for gene and glycans also. So how do we decide that how to what extent we should showcase the parameters in a database? So if you can tell this question.
So when you say parameters, means like in terms of search parameters, or filtering parameters?
So it's about like what are the parameters that you want to showcase in your database, basically.
So I can think of parameters. When I think of parameters, I'm thinking about what kind of information. So, you know, the mass of the biomolecule, the classification. Any annotation. So I think all annotations, or most of the annotations, can be considered as some parameters, some form of parameters. They might be numerical, or they might be just annotation, text annotations. What to show in a database also is key. And I think I had one slide where I was trying to show that in GlyGen, there are other resources which take a different approach. I'm not saying that our approach is the only approach, where when you're building a glycoinformatics or a glyco-related database, you can ask the question, let's put everything glycan related in there, right? Make it as comprehensive as possible. The other way is that, no, you try to put things, because no biomolecule works on its own. You try to connect it to different data sets and integrate it so that people can ask the systems level questions. So I think from a GlyGen perspective, we are interested in that ability of people to come inside the resource and ask the type of questions they will ask, and stay in the resource to really be able to answer biological questions, which they can try to go and see experimentally validate. So that's how we are looking at the different types of parameters that we can add in a resource. Does that answer your question?
Yes, sir. Thank you so much for just taking my questions. And since we are taking a lot of your time, and so I would like Dr. Manish Kumar, who is the faculty advisor of RSG-India, to just give her a conclusion and his insights from this session, so that we can hear from him. Hello, Dr. Manish.
Hello. Am I audible?
Yes.
Yes, sir, you are. Yes, sir, you are audible.
Yeah. Okay. So this is Manish Kumar from University of Delhi South Campus. And first I would like to thank Professor Michael and Professor Raja for taking time to brief us about their databases. So I think personally, I have learned a lot with this talk. So I have never appreciated the role of glucose molecules in our system. Mostly when we teach students also, we teach about that diversity is brought about from the amino acid, different addition of amino acid in the protein. But we never appreciated importance of glycan. Okay. I worked on autoimmune diseases also, but it never occurred to me that glycan molecules can be responsible for that. So thank you for giving us this insight. And from Professor Raja, one thing I was very delighted to hear that bioinformatics is not about informatics only. So I always say in my lab that, look, we are doing biology, we are not doing informatics. So always try to see the results that you are getting in terms of the biology. And I request every students who are interested in bioinformatics that look, whatever results or whatever problem you are taking in light of biology, not in that I know this programming language, I know this programming language, I can do this, I can build this database. So, okay, these are essential skills, but these are not the primary skill that you need to be successful in bioinformatics. So I hope a student will also appreciate this fact. And again, I will thank Professor Raja and Professor Michael for taking time to give us this insight about the glycan molecules. And definitely I will write to Michael and Raja about some project that I thought that, okay, with our autoimmune work, we can also do this type of thing. So definitely I will write to you sometime soon. Okay, thank you. Thanks a lot.
Thank you, Dr. Kumar.
And,
Yes, of course, please be in touch, and everybody on the call today, feel free to email us.
Yeah, and thank you everyone for attending this talk. So it's quite late in India. Thank you.
Thank you, Dr. Manish, for taking your time and just joining for this session. So, on behalf of RSG India, I would like to thank Professor Raza as well as Michael for taking your time, such precious time from your schedule, and letting us know that how important the glycans are. Like in one of your slides, I could see collagen and hyaluronic acid, which is a very popular thing in today's skincare also. So I think most of the students have just got to gain interest from that thing also. So thank you for taking your precious time and just letting us know that how important this concept is, other than like DNA and RNA only. So thank you so much. And I would really like to thank RSG India also for just coming up with this platform and letting us with these brilliant bioinformaticians among us, so that we can learn and excel in our career and can implement these things that we have learned from these sessions. So thank you, RSG India. And I would also like to thank the whole team of RSG India, as well as our volunteers, Arushi, Shreya, Neelam, Girika, and Sabyasachi, for putting a lot of effort in building this type of sessions successful. So thank you, everyone. So don't please go through our website and look at our upcoming events, and please stay connected with us in upcoming sessions, because we have so many things like this coming up in next days. So thank you, everyone. From here, I would just like Akriti to conclude the session.
Yeah, thank you, Nikita, and especially thank you, Professor Mazumder and Professor Tiemeyer, for your time. If you would please allow us to take a picture, so that we can take a group picture. Can we please turn on our cameras for a group picture? And thank you, Dr. Kumar, for inspiring all of us to see the bio behind bioinformatics, not behind, in front. And yes, it was a great session, and definitely we will have more questions in the feedback form. So I request all of you to please fill the feedback forms, and if you still have any questions, you can write to us, and we would be happy to convey them to Professor Mazumder and Professor Tiemeyer, and whenever they have time, they can answer. I request Govinda to please stop sharing the screen, I think, and then we can take a picture and conclude the session. For whoever it is possible, please turn on your cameras quickly to.
Yes, it's done, Akriti. I've done.
Yeah. So yes, thank you, everyone. And in the interest of time, we are leaving some questions. Those were the questions which are left unanswered. We will try to email them to both the professors. And thank you, Professor Mazumdar, and thank you, Professor Tiemeyer, for starting your morning, Friday morning, with this session. We hope it was good for you. We are going to have a very great sleep with all this wisdom and learnings. And yes, we can all work together in this. I hope glycans become a part of central dogma soon, so that we have in our next textbooks about this. And we will definitely get some good interns from India soon. And thank you so much.
Thank you, everybody.
Yeah. Sir, Manisha, are you saying something? Okay. So yeah, thank you everyone for joining.
Bye, everybody. Thanks.
Bye-bye.
Bye, sir.